Okay, welcome to Chapter 4. In this chapter, we're going to study the composition of tissues. We'll be talking about their anatomy, their function, and where they're found in the body, and we'll try and define their roles for you as well. Now, individual body cells are specialized. Each cell type performs specific functions that help keep the body's internal sense of balance, which we know as homeostasis. Tissues are groups of cells that are similar in structure and perform a common function, and the study of tissues is what's termed histology. Now, before we delve any further into a discussion of the different tissue types that are found in the human body, we should probably familiarize ourselves a little bit with where a lot of these tissues come from. So we have to start all the way back here at the beginning and consider now this idea of um, fertilization, okay, where what we'll have is an egg and a sperm. Remember that each of these are haploid cells, and what they will do is they will meet in the process of fertilization and form a diploid cell that's called a zygote. Okay, so zygote, ZYG, right there. And immediately what will happen is that this zygote will begin to multiply very, very rapidly. One cell will become two, two will become four, four will become eight, and so on, until we form a little cluster of cells that looks a little bit like a raspberry under a microscope, and this cluster of cells is called a morula. Okay, morula. As this cluster of cells moves down the fallopian tube towards the uterus, a very interesting thing will happen, which is that there's a sort of a tough membrane that surrounds the morula. It will open up and hatch, and what you'll generate is a fluid-filled ball called a blastocyst. And so if we were to take a coronal section through the blastocyst, what we would find is a layer of cells, and then inside that layer of cells, clinging to one side of this fluid-filled ball, would be the inner cell mass. And the inner cell mass is eventually going to be the embryo. And so if we were to draw that, it would look a bit like this. Okay? So these little red cells right here, those are eventually going to be the new human being. If we were to take this inner cell mass and zoom in on it in the weeks following the production of the blastocyst, let's indicate here blastocyst so we know what we're talking about. That's the blastocyst, that whole thing. We could take a transverse section through the blastocyst, and what we would find is a layer of cells, which embryologists would call the ectoderm, and then another layer of cells below it, which embryologists like to call the endoderm. Okay? So the endoderm here in blue and the ectoderm here in red. The next event that'll follow will be the production of a tiny groove in the middle of the ectoderm, which is eventually going to become the spinal cord and the vertebral column, but for right now we're just going to call it the primitive streak. So let's draw that here. So the primitive streak looks a bit like this. So just a little groove right here. And then the endodermal cells are underneath it. Here. And then within the population of ectodermal cells, what will happen is that certain cells will start to migrate through the primitive streak and populate this liquid-filled space right here, and we call these cells mesodermal cells. So let's draw a few of them in black here. Okay, so they kind of pop out of the population of the ectoderm, and then they move through here, and then they fill this little fluid-filled space. And so what we end up with now is a disc that's got 3 layers of cells. The middle layer is the mesoderm. The upper layer is the ectoderm, and the inner layer is the endoderm. Okay, so let's fill these in. Endoderm here in blue. Okay, and ectoderm here on the top in red. With the primitive streak still there. Okay, and so those are the 3 germ layers. We'll label them ectoderm, ECT, and then mesoderm, mesoderm, put a little arrow there, and then underneath endoderm. Now the significance of this event is that these 3 populations of cells are eventually going to form all the other tissue types in the body. The ectoderm is going to produce the skin and the nervous system. The mesoderm is going to produce the cardiopulmonary and the musculoskeletal system. And the endoderm is going to form the linings of a lot of the internal organs and also a lot of the membranes that support those organs. And the significance of this event is that once these 3 cell populations have been established, they can't go back and be something else. And the reason for that is that the information in their nucleus has been changed in a way that even though the information is still there, the mesodermal cells can only read from the mesodermal segment of the DNA, the ectoderm can only read from the ectodermal segment, and the endodermal only from the endodermal segment. And as these cells continue to mature and generate their adult tissue types, the information that they're going to be allowed to access in the nucleus is going to become more and more restricted. And this is going to shape the way these cells look and act, and eventually the tissues that they form and what those tissues do. So, if we were to group the different primary tissue types that we're going to cover in this chapter, they include epithelial tissue, which covers the insides of organs, the outer surfaces of the body, provides a lot of protective barriers, also can act in diffusion and absorption. Connective tissue, which is the body's glue, it helps to support and reinforce other tissues. And muscle tissue, which is capable of contraction, so it can produce movement. And then finally nerve tissue, which is one of the body's 2 methods of communicating with itself. The nerve tissue is specialized for sending and receiving electrical signals. And you can see these different tissue types here. The nervous tissue you'd find in the brain, the spinal cord, which are parts of the central nervous system, and the peripheral nerves. Muscle tissue you find in skeletal muscle, which is attached to bone and produces voluntary movement, in the heart, where it allows the heart to pump blood as cardiac muscle, and in the walls of hollow organs, blood vessels, and lymphatic vessels in the form of smooth muscle, where it produces internal movement. Epithelial tissue forms a lot of different boundaries between the inside and the outside world for the body. It protects and secretes. It also absorbs and filters liquids as well as gases, depending on its location. We find it in the lining of the digestive tract and other hollow organs, and we also find it on the skin surface. We find it in the lungs. We find it in a lot of places. Connective tissue is found virtually everywhere in the body, and it's the body's reinforcement and glue. It supports, protects, and binds other tissues to each other so that they can withstand physical and chemical force. Examples of connective tissue include bone and cartilage, tendons and ligaments, adipose tissue, which we call fat, and what we call connective tissue proper, which is found primarily in the dermis, which is just beneath the epidermal layer of the skin, and also in the fascia, which is connective tissue that holds muscle fascicles together so that when muscles squeeze, they don't come apart. And we'll look at each of these tissues in a little bit more detail as we move through Chapter 4. Now, when we study human tissue, what we have to understand is that we need to treat it in order to be able to visualize some of the internal anatomy. And the way that this is done is that the living tissue is sampled, for instance, in the form of a biopsy, or perhaps even a piece of tissue that was removed from an organ after an individual has been deceased. That tissue is then thinly sectioned and is fixed so that it won't decompose. This is essentially with a preservative. Examples of preservatives include compounds like paraformaldehyde. And then we cut the tissue in extremely thin sections and stain it in order to allow us to see the structure inside the cell. Allows us to visualize the nucleus, the organelles, and a lot of the intra and extracellular proteins. And this gives us an idea of what normal tissue looks like. One of the reasons that we engage in this type of histological study is to compare tissue taken from a patient that may be suffering from a disease state and compare it to normal tissue and look for any differences in cell shape and in the components that we see inside the cell. For instance, the appearance of abnormal inclusions or abnormal structures in the cell, or a change in the cell shape, could potentially indicate a disease state. Now, epithelial tissue is the first tissue type we want to address, and the bottom line with epithelial tissue is that what distinguishes it from most other tissue types is that it likes to form boundaries. When you look at epithelial tissue, the number one characteristic that jumps out at you is that a lot of cells are packed very closely together, and there's very little extracellular space between the cells. There's two main types of epithelial tissue, and they're classified by their location. We have epithelia that cover or line internal or external surfaces, and then we have epithelia that are responsible for secreting components. Examples of this type of tissue include glandular epithelia, such as you might find in a sudiferous or a sebaceous gland, or in an endocrine gland of some type. The main functions of epithelial tissue include protecting the body. For instance, the epithelial tissue of the skin is a protective layer against infection. It also prevents water loss, and it also serves as a thermoregulatory organ, and it also provides an area of synthesis for vitamin D, which helps mineralize bone. That's just a few of the roles of the skin and the epithelial tissue's role in the skin, which is the upper layer of the skin. It's also important in absorption, in filtration, in excretion and secretion, and it's also going to have a role in sensory reception via its close association with components of the nervous system, which are called sensory receptors. It can respond to stimuli in the external environment, such as sound or touch or pressure or pain or tickle or changes in temperature that we can sense. And as a result, this allows us to act on our environment in order to ensure our survival. Some characteristics of epithelial tissue include polarity, specialized contacts, the support that it has from the underlying connective tissue, and the fact that it is often avascular, meaning it tends to lack blood vessels. However, it is innervated by the nervous system, again, serving its role as a sensory organ, both to the external environment, but we should also point out that there is a lot of innervated epithelial tissue inside the body that is utilized by the autonomic nervous system to report on our internal functions, which we may not necessarily be consciously aware of, but are still important in maintaining our health. It also has a high mitotic index, meaning that it can regenerate when cells wear out or when cells are damaged, and that's very important because if epithelial tissue didn't have this ability, then after a very short period of time, as cells wore out, were sloughed off, or were destroyed as a result of interaction with stimuli, then we would lose those protective coverings. So let's talk a little bit about this idea of polarity. Okay, epithelial cells have an apical surface, which is the part that's exposed to a body cavity, say to the internal opening of an organ or onto a free surface of some type, and then a basal surface, which is generally attached via the basal lamina to connective tissue or some other tissue types. Both of these surfaces are going to differ in their structure and their function. If we were to draw, say, a typical epithelial cell, let's use black here, we could define these different regions. So let's just say that this is an epithelial cell in question, okay? And this part up here would be the apical surface that I'm drawing right now. And this would be the nucleus, okay? And the DNA would be in there. And then this would be the basal surface. Okay, so there's the basal surface. There is the apical surface. And then you've got the lateral surface, which is here and also over there. These are the sides of the cell, so lateral. Now, what's the significance of these different surfaces? The apical surface you could think of in many cases as the business end of the epithelial cell because it's often modified for absorption or secretion. The microvilli, which are these little tiny fragments, these little extensions at the apical surface of the cell, are an example of a surface area modification that lets the cell do a lot of absorbing things and secreting things in a very small physical space. And also, we've often find at the apical surface membrane proteins that are specific to that cell's function. Things like channels or pumps are often found on the apical surface where they might be absent or might be quite different on the basal surface. On the basal surface we might find cell junctions that are designed to attach the cell to the basal lamina so that it doesn't basically fly off due to mechanical or chemical stress. And there might not be the same types of transport membrane proteins on the basal surface as the apical surface. On the lateral surface, we often find junction proteins that allow the epithelial cell to basically hook up with its neighbors on either side. Examples include tight junctions and desmosomes, and then in some cases we'll even have communication networks between neighboring cells, which are like tunnels that things can travel through going from one cell to the next. So that's the significance of these cells, is that they have sides, and the sides aren't identical. The sides differ in their composition and in their function for the cell. The apical surface may be smooth and slick. Many of them will have microvilli, which are there to, again, increase the surface area of the cell so that it can do its job more effectively. An example is the brush border of the small intestine, where the macronutrients are chemically broken down into small enough molecules that they can be transported across the epithelial lining of the intestine into the bloodstream, where they can be eventually transported to the cells that need it for energy. And it's those microvilli that increase the working space that these cells can use in order to do that job. In addition to that, these microvilli also allow lots of secretion to go on. In order to chemically break down the macronutrients, we have enzymes that are embedded in the microvilli, or in some cases secreted by the microvilli, that chemically break down our macronutrients, our complex carbs, our fats, and our proteins into their smallest building blocks so that they can be absorbed into the bloodstream. Other secretions include mucus, which is important in lubricating the food as it slides through the digestive tract and also protecting the epithelial tissue from abrasion. Okay, so again, the microvilli increasing that surface area and helping in all those tasks. Sometimes you'll find cilia on the apical side of an epithelial cell, and what cilia do is they move, and they generally move a liquid across their surface from point A to point B. An example would be the pseudostratified ciliated columnar epithelium of the trachea. Bottom line, if we were to look at these cells, they'd look a little bit like this. We'll draw them in black again. They're tall, okay, and then at their surface they have these hair-like extensions that use ATP to produce movement, and the movement that they produce, this would be the nucleus of the cell, is to slide mucus up out of the lungs and into the oral cavity, where you can either swallow it or you can spit it out. And the bottom line is that the mucus traps particulates that we don't want to have end up in the lungs, where they would produce irritation and inflammation. Okay, so there's our apical end of our ciliated columnar epithelial cell, and then this would be the mucus that it's propelling in this direction, and this would be towards the mouth, okay, and out of the lungs. And that helps keep the lung tissue from scarring and inflaming. So, very important, these extensions on the apical surface. The basal surface of epithelial tissue is connected to an extracellular matrix component called the basal lamina, which is basically glycoprotein and collagen that's adjacent to the basal surface. It forms a sticky sheet, and also in some cases provides a filter through which components can move. It also serves as a scaffolding for cell migration and in wound repair. A lot of times when we damage tissue, this basal lamina serves as a chemical breadcrumb trail for cells that need to get to the area of the damage in order to remove dead cells, as well as a signal for the cells near the area to try and regenerate new tissue in order to seal the wound off. Okay, so basal lamina, some important stuff, and it marks the end of the epithelial layer. Okay, it is an actual epithelial product. Okay, specialized contacts that we find in epithelial tissue allow them to fit closely together so that they can form sheets that don't come apart, so that allows them to resist shear stress. These specialized contacts that we find are often on the lateral sides of these cells, and they include tight junctions and desmosomes, which you could think of basically as spot welds, essentially a nice seal between neighboring cells so that they don't come apart under physical or chemical stress. The tight junctions and desmosomes are particularly important in the stratified squamous epithelia of the skin because they provide a barrier against infection as well as a barrier to moisture. And as a result, this allows the body to control its fluid balance. The only way that fluid gets into the body is through the digestive system via the mouth. If we didn't have these waterproofing junctions in the skin, then every time we took a shower or stood out in the rain or took a bath, we'd soak up water like a sponge, and that would interfere with our body's homeostasis because it would screw up our fluid balance and our electrolyte levels. Okay, there's connective tissue support in epithelial tissue. This provides an underpinning that resists physical and chemical force. There's a reticular lamina that lies under the basal lamina that's made up of primarily collagen. And then there's a basement membrane, which is the basal lamina and the reticular lamina fused together, that reinforces the epithelial sheet and resists those physical forces we were talking about. And of course, it also defines the deep boundary of the epithelial layer. Okay, so this is again a reinforcement of the connective tissue's role as your body's glue. Hold stuff together, stick stuff together. Epithelial tissue is avascular, but it is innervated, meaning there are no blood vessels that we see in epithelial tissue, so it's nourished primarily by diffusion from underlying connective tissue, but it is innervated by nerve fibers, and this helps the skin and the epithelial tissues serve their role as a sensory organ. Epithelial tissue has a unique ability to regenerate itself when cells wear out or when they're damaged, and so they have what we call high mitotic index. They're stimulated by the loss of apical basal polarity and lateral contacts, meaning basically when an epithelial cell loses one of its buddies, either on top of it or next to it, then it's stimulated to divide in order to replace that missing neighbor. Some are exposed to friction, and some are exposed to hostile secretions, and as a result they frequently get killed and have to be replaced. If adequate nutrients are around, we can replace lost cells as a result of the division of neighboring cells or even deeper cell layers in order to repair the damage that results from these day-to-day wear and tear kind of events. So we can classify epithelia by their layering and by their shape. All epithelial tissues can be grouped this way. Simple epithelia are simply a single layer of cells, while stratified epithelia means that we have two or more layers of cells. And the shapes can change in different layers. We can name these cells according to their different shapes. Squamous cells are flat like a pancake, cuboidal cells are box-shaped, and columnar cells are taller than they are wide or deep. When we name stratified epithelia, we classify them by the shape of the cells that are in the apical layer. So in these two pictures here, we're looking at simple squamous epithelia and stratified squamous epithelia. Squamous because the cells are flat like a pancake, simple in this case because there's only one layer. Here, stratified squamous because there's more than one layer of epithelial cells, and the apical surface is made up of squamous epithelia. Okay, so that's one of the ways that we name these tissues. This is just showing you the different shapes that epithelial tissue can take on. Squamous cells shown here, flat like a pancake. They look scale-like. The nucleus is flattened, while cuboidal cells are box-like with a round nucleus, you can see here. And columnar cells are tall, have an elongated nucleus, and we can find any of these cell shapes in different tissues in the body, either in the stratified or the simple conformation. And we'll see some examples of those as we move through Chapter 4. Simple epithelia are designed beautifully for absorption, secretion, and filtration. They tend to be very thin, and the reason for that is that this provides a minimal barrier to the movement of molecules via diffusion across this layer. And an example of a tissue that is simple epithelia would be alveolar tissue, and you're actually looking at some alveolar tissue here. This is a micrograph of cross sections in the alveoli. These are the delicate sac-like structures that are found in the lungs that facilitate gas exchange. Essentially, when you breathe, the oxygen in the air that you inhale diffuses across these very thin sheets of simple squamous epithelia and into the blood that's coming to the lungs from the heart. At the same time, the carbon dioxide that's in that blood is diffusing out of the blood and into the air that you're going to exhale. The result is that the blood will load up with oxygen, head back to the heart, and the heart will then pump it to the rest of the body, providing oxygen that the cells can then use in order to make lots and lots of energy so that they can live. What you're seeing here is just sort of a cartoon of what simple squamous epithelia looks like, and this, of course, is the micrograph. You can see that the cells have a flattened lateral shape, very little cytoplasm, and they function where diffusion is important. The lungs are shown here. We also find these types of cells in the kidneys. The kidney's job is to clean the blood, and as a result, there's a lot of diffusion going on in the kidney tubules, and so we find this tissue in the kidney tubules at certain points along the structures in the kidney that actually do the blood filtering. They're called nephrons. And again, you can see perfect example of how form follows function. If we interfere with the structure of this tissue by putting a layer of crud on it or by having the tissue scar, then its function is going to be impaired. For instance, if lung tissue scars, it's very difficult to load oxygen into the blood, and as a result, for those tissues to generate the ATP they need in order to live. So again, tissue perfectly designed for its function. Okay, simple squamous epithelia is also found in endothelial layers. These are found in the linings of the heart and the blood vessels and the lymphatics, where they serve, again, as a boundary layer between the fluid that's inside the organ and the outer layers. Mesothelial tissue is essentially lining the serous membranes of the ventral body cavity. They produce a slippery fluid that lubricates the internal organs so that when they move, they don't rub across each other and produce a lot of undue friction and inflammation and scarring. So our first tissue that we want to look at after simple squamous is simple cuboidal. This is a single layer of cells important in either secreting or absorbing functions in the body. It forms the walls of the smallest ducts of the glands and also is found in the kidney tubules. This is a cross section actually through kidney tissue, and you can see this single layer of box-like cells that lines the kidney tubules. This is what it would look like in cartoon form, and this is an actual electron micrograph. And again, remember that the kidney's job is to clean the blood, and so a lot of secreting and absorbing goes on in the kidney tubules. And here again we see an example of form following function. These are the nuclei of the cells, and then if you look real close, like that would be a cell, that would be a cell, that would be a cell, and this is the lumen of the tubule. This is where the fluid flows through. Long story short, if you're wondering, essentially what the kidney does is it removes toxins from the blood, things like ammonia and urea, stuff that if it were allowed to build up in body tissues would change chemical conditions and cause cells and tissues to die. Those toxic compounds end up in the urine. At the same time, these kidney tubules also put useful solutes back into the blood, things like water and electrolytes, and also fuel in the form of things like glucose and amino acids, so that the cells can eat. And so that's a big task, and we need a tissue that's good for it, and hence we see this simple cuboidal epithelia in the kidney tubules. Simple columnar epithelia is a single layer of tall, loosely packed cells, again designed primarily for absorption and secretion. This is an actual micrograph taken from the epithelia of the small intestine. Notice that we have a couple of different cell types here. These are the simple cuboidal epithelial cells that I'm touching here, and then these are goblet cells, and what they do is make mucus, which helps to protect the epithelial lining of the intestine and lubricate the food as it slides through the intestine, um, and also again helps to protect these cells. Again, these cells are going to be subjected to physical force, friction, and the like, and the mucus helps to minimize that. Okay, so it's a lubricant and a protective layer. Um, Notice that the columnar cells on their apical surface here have a brush border, and this brush border is essentially microvilli protruding from the apical surface, and that's again designed to increase the surface area of this tissue type so that it can absorb and secrete, which are vital functions for digestion. You could think of the microvilli as expanding the working space that these cells need in order to absorb nutrients and pass them into the blood, and also secrete components that help break down the macronutrients that are in the digestive system in order to get them into the blood in the first place. We have to chemically and mechanically break down the macronutrients in order to get them into the bloodstream, and we do that in part as a result of the enzymes that are either secreted by or embedded in these microvilli that are on the apical surface of these tissues. Pseudostratified columnar epithelium has cells that vary in height. It's a simple epithelium. These cells down here are actually short, and the cells that neighbor them are tall. The nuclei appear to be at different levels, so it looks as if it's a stratified tissue, but it is in fact a simple tissue. The role of this tissue is primarily secretion and absorption. You can find goblet cells if you look very closely. You can see a few of them here and here, so on. They make mucus, and the mucus in this region, this is a piece of tissue taken from the upper human trachea. The mucus is used to trap particulates and pathogens so that they don't get into the lungs, and then these cilia use ATP to produce movement, and that helps beat the mucus up towards the mouth where we can either spit it out or swallow it. The bottom line is it doesn't end up in the lungs where it would cause irritation and inflammation and scarring. Stratified epithelial cells are going to have two or more cell layers, and they'll regenerate from the deeper layer, which we could think of as an adult stem cell population, as the upper layers are sloughed off and need to be replaced. They're more durable than simple epithelia simply because there's more layers there, and so they can withstand more physical force. And they're primarily for protection. And here you can see an example of simple or stratified squamous epithelia. This is one of the most widespread of the stratified epithelia. The free surface is squamous, and so that's why we call it stratified squamous. Notice the flattened cells up here, and then as we move to the deeper layers, they kind of look cuboidal a little bit, but because the apical surface is flat, the whole thing is considered stratified squamous. It's located where wear and tear is going to happen. This is a very tough tissue, so it resists that sort of mechanical stress. The cells that are furthest from the basal layer are the ones that are going to be shed and then replaced by the basal layer cells as they divide rapidly and repopulate these upper layers. This is actually a slide taken from the esophagus, and that's the food tube that connects the mouth to the stomach. And as you can guess, it has to withstand a lot of physical force. As the food goes from the mouth to the stomach, it rubs past these layers. There is a layer of mucus that helps to lubricate this tissue, but even at that, there's going to be physical force, and at some point the apical cells are going to die and have to be replaced by the cells that are generated by the deeper layers. Stratified columnar epithelia is rather rare. We find it in the sweat and in the mammary glands. They're usually about 2 cell layers thick, and they're limited in their distribution in the body. We see a little bit in the pharynx and in the male urethra, and some in glandular ducts. They also occur at the transition area between other types of epithelia, where only the apical layer is columnar. Now, transitional epithelium is special because it has the ability to stretch and then recoil. We find this lining hollow urinary organs like the ureters and the bladder. The basal layer is cuboidal or columnar, and it has the ability to change shape depending on how stretched out the tissue is. If the tissue is real stretched out, the apical layers will look squamous, and if the tissue isn't really stretched out, the apical layers will look cuboidal or columnar. But again, it's the presence of elastic fibers in the cells and between the cells that allow this unique ability to basically stretch out and snap back. And again, form follows function here. As the urine courses through the urinary system, we want distensible structures, things that can stretch out as things fill with liquid in order to accommodate that liquid and eventually serve to void it from the body. If this tissue wasn't distensible, then the frequency of urination would go way, way up. Okay, glandular epithelia is primarily tasked with secreting. It makes a product inside the cell that then gets transported across the cell membrane and then into the outside world. These secretions are generally aqueous components. They're classified by the site of the product release. They can either be endocrine glands, meaning that they have a duct, or endocrine glands, meaning they don't have a duct, or exocrine glands, meaning that they do have a duct. And so let's reinforce that here. Let's in red. Endocrine glands are ductless. This is a very important distinction. And exocrine glands have ducts. Okay. They're also classified by the number of cells forming the gland. The glands can be unicellular, meaning the entire gland is one cell. An example of that would be the goblet cell. Or they can be multicellular, such as in sweat glands or oil glands or mammary glands. Endocrine glands are ductless. Their secretions are not released into a duct. Instead, they are actively transported across the cell membrane and directly into the bloodstream, often by exocytosis. An example of a ductless gland would be something like the adrenal gland or the pituitary gland. And if we were to sort of draw you a mock-up of how this would work, essentially this would be the glandular tissue, and in endocrine glands, the glandular tissue is going to have a lot of blood vessels coursing through it because the products that it makes has to be very, very close to the blood supply in order for them to travel throughout the body. Okay, so this would be the endocrine glandular tissue. And then we would have blood vessels running all through this. We'll show those in red here. So these are our blood vessels running all through the tissue. And then these blood vessels would exit the gland and, of course, run all throughout the body. And then we would have target tissue, and we'll just indicate the target tissue here in magenta. And what defines a target tissue for an endocrine gland is the presence of receptors for the hormone product that they make on the cell surface. So this is our target tissue. And then these little receptors would look just like, act just like little catcher's mitts for the chemicals that are secreted by the endocrine gland. And so they would be on the surface of the target tissue. And then the hormone, which is actively secreted into the blood, we'll show here in blue. And what happens is that hormone just gets squirted straight into the bloodstream, and it travels down the bloodstream. This is our direction of travel here. And then when it gets to its target tissue, it binds to the receptors. Okay? And when it does that, it causes a change in the target tissue's behavior. The target tissue might divide, it might make a special product, it might die. It all depends on the nature of the chemical signal coming from the endocrine gland. And then the conditions that are created as a result of that interaction with the hormone will then feed back to the original endocrine gland and tell it to quit producing that secretion, and that's what we call negative feedback. That's one of the most important ways that homeostasis is maintained in the body, is through this negative feedback relationship. So the target organs respond, and the negative feedback loop is completed. Exocrine glands have secretions that are released onto body cavities or onto body surfaces. They're more numerous than endocrine glands, and their products are secreted into ducts. Examples include mucus, sweat, oil, and salivary glands, mammary glands. Here you can see an example of the only unicellular exocrine gland. This is the goblet cell. We find these in epithelial linings of the intestinal and the respiratory tract. They make a product called mucin. That's what you see in these little vesicles here. And it dissolves in the water, forming mucus, which is a protective lubricating coating. In the upper respiratory, of course, it serves to trap particulates so that they don't end up in the lungs. And in the digestive system, it's a lubricant for the food as it slides through the digestive pathway. And it also protects the epithelial cells from undue physical stress and strain, so that they can do their job more effectively and last a little longer. Multicellular exocrine glands are made up of a duct and a secretory unit. They're usually surrounded by connective tissue, and they supply blood and nerve fibers. They extend into and divide the gland into lobes, which can be grouped according to their structure via branching and as well as their shape. And also they can be grouped by their type of secretion. If we look at their structure, you can have simple glands, which are going to have unbranched ducts. Okay, so you can see here an example of some simple glands here, okay, with unbranched ducts. Or you could have compound glands, which are going to have branched ducts. Okay, so there's an example of a compound gland. Okay, there's another compound gland. These are simple. Okay, and they can be differing shapes. The cells can be tubular. The ducts can be tubular, alveolar, or tubulo-alveolar. If they're tubular, they're tube-shaped. So it's just like a blind tube. If they're alveolar, they're going to have kind of a bubble end to them. And if they're tubulo-alveolar, then they're going to have a portion that's elongated like a tube, and they'll terminate into this bubble-type shape. And then they're going to differ by their secretion type. Most exocrine glands are merocrine, meaning they secrete their products by exocytosis directly through the cell membrane, and then that product moves through the duct onto the surface of the tissue or into the lumen of the organ or vessel. Apocrine glands accumulate products within only the apical portion of the cell, which then pinches off and ruptures, and that becomes the product. We think that there may be some glands in the epithelia that operate this way, where holocrine glands accumulate products within them, and then the entire cell ruptures and becomes the secretory product. Okay, so you can have different shapes shown here, and then you could have different secretion types, and then all combinations of this are possible in living tissue. Okay. All right, that brings this portion of Chapter 4 to an end. Again, review this material because it will be covered on upcoming exams and tests, and I'll see you in class. Okay, now carrying on in our discussion of living tissue, we want to talk next about connective tissue. It's one of the most abundant and widely distributed of the tissues we're going to talk about, and the reason for that is that it has a very important job, which is to provide structural support and essentially act as a glue or a cement for body tissues. This is what allows tissues of different types to be attached to each other. There's four main classes: connective tissue proper, cartilage, bone, and blood. And if we look here at our first example of connective tissue, this is connective tissue proper. It can be broken down into a couple of different subclasses: loose and dense connective tissue, which basically refers to the organization of the extracellular matrix, which is essentially the job description for connective tissue. Loose connective tissue, it can be broken down into either areolar connective tissue, which you find in the dermal layer under the skin and between muscle fascicles, where it's known as fascia. Adipose connective tissue, which is basically fat tissue. And then reticular connective tissue, which you find in certain organs in the body, and we'll address exactly where they're located when we get to that point. Dense regular connective tissue is found in tendons and ligaments. Irregular connective tissue has a more disorganized extracellular matrix, and elastic connective tissue, such as you find in the epiglottis and in the external ear, has the ability to stretch and contract. Cartilage is another very important type of connective tissue. It can either be hyaline, elastic, or fibrocartilage. Hyaline cartilage, for instance, is found at the ends of bones in articulations that we call diarthrotic joints. Those are the freely movable joints. Elastic cartilage, again, found in the epiglottis and in the external ear. And fibrocartilage is found in, among other places, the intervertebral discs, where it forms the outer surface of the disc, which is called the annulus. The cells that you find in these kinds of tissue, for loose connective and dense connective tissue, fibroblasts are what actually produce the matrix and remodel it. Fibrocytes are the cells that become trapped in the matrix once it's produced. And then you'll also see lymphatic tissue and connective tissue, and again, the purpose for those cells is to defend against infection. Adipocytes are basically fat cells, and you'll find them obviously in adipose tissue, but you may also occasionally find them in some of the other connective tissue proper types. You can see here a description of the matrix. It's essentially a semi-solid substance, contains a lot of hyaluronic acid and chondroitin, but it also contains quite a bit of water. And you'll also see collagen and elastin and other types of protein fibers running through this extracellular matrix to provide strength. General features are listed in the final column over here. I won't go through those right now. We'll get to them when we actually hit these tissues. If we look here at cartilage, the chondroblasts are the cells that actually generate the cartilage extracellular matrix. This is particularly true in growing cartilage, and the chondrocytes are the cartilage cells that become trapped in the matrix. You can see them here inside these little chambers that are called lacunae. Essentially what they've done is they've sealed themselves in, and the only way they can get nutrients is through diffusion of synovial fluid through the extracellular matrix which allows them to continue living and maintaining the matrix. Um, the fibers that you're going to find in cartilage, again, a lot of ground substance here, so a lot of hyaluronic acid and chondroitin sulfate. You'll also find a lot of collagen and elastin in cartilage tissue as well. Okay, moving over to a tissue you're probably more familiar with. This is bone. Comes in two flavors, compact and spongy bone. Compact bone is found on the outer surfaces of bones. It's very tough and dense, and as a result, resists compression force, but it also features quite a bit of protein, which is resistant to shear stress. Spongy bone lies deep to compact bone and lightens the bone, but it also serves to distribute applied force so that it's less likely that the bone will crack when it experiences a physical trauma. The cells that maintain bone are the osteoblasts, which are the depositors of the hard matrix, which is mostly calcium carbonate and calcium phosphate, and the osteocytes, which you can see here, which are what the osteoblasts become once they've been trapped inside that matrix. And the way they get their nutrition is through cytoplasmic extensions, which radiate out from the cell and touch the cells that are close by in these regular depositions of dense matrix that are called lamellae in compact bone, and simply to neighboring cells that are embedded in structures that are called spicules in spongy bone. The matrix is again a gel-like ground substance, but it's calcified with calcium phosphate and carbonate. And there's also a considerable amount of collagen in bone. If you want to demonstrate that to yourself, the next time you get a bucket of chicken, save the wishbone and dunk it in cider vinegar for 2 or 3 days, and the calcium salts will dissolve away, and all that will be left is the protein matrix of the bone, and that wishbone will snap and bend just like a rubber band will. Blood is one of our liquid connective tissues. Lymph is the other one. And it's made up of plasma and formed elements. The plasma is the liquid component of blood. It contains proteins and waste products and electrolytes and also a considerable amount of water. And the formed elements are the erythrocytes and the leukocytes and the thrombocytes. The thrombocytes are important in blood clotting. The erythrocytes carry oxygen and carbon dioxide, as does the plasma. And the leukocytes are basically our defense against disease. The formed elements are made primarily in the red bone marrow, which is found in the flat bones of the human skeleton, and are continuously regenerated and recycled. Um, the liquid matrix has no fibers in it. There are, of course, a lot of proteins in the plasma that are important in processes such as blood clotting, as well as in defense against pathogens. The proteins that do this in the blood plasma are known as antibodies, and also their associated defense proteins, which we call complement. So what are the major functions of connective tissue? Well, we've talked about a few of them already, right? It binds the body together and provides a support framework. It protects a lot of stuff. It protects us from infection and disease because it provides a barrier. It also physically protects a lot of our internal organs. The skeletal system, for instance, physically protects the brain and the spinal cord, as well as the heart and the lungs. And the pelvic girdle protects the bladder. And of course, the underlying tissue, the dermis, is a barrier against infection by pathogens because it provides now a tough layer for the pathogens to penetrate, but it's also populated by lymphatic tissue, which will attempt to destroy any pathogen that makes it through the upper layers of epidermal tissue. It's also an insulator, primarily in the form of subcutaneous fat and the fat that's found around organs. This pads and protects the body, but it also helps to hold in heat, and it also serves as a fuel store. And it also is important in transport, and here we're thinking primarily of the lymph and the blood as the liquid connective tissues. 3 characteristics that make connective tissue different from other tissues is that they have mesenchyme as their origin tissue, which is a derivative of mesoderm. If you remember, back to the beginning of the talk and the establishment of the three primary germ layers, the ectoderm, the mesoderm, and the endoderm, the mesoderm eventually generates the mesenchyme. They have varying degrees of vascularity, which means that depending on the connective tissue we're talking about, you'll see more or fewer blood vessels. And they have an extracellular matrix, and this is probably the most important feature of connective tissue because the extracellular matrix essentially defines the connective tissue. Connective tissue is not composed mainly of cells. It's largely non-living extracellular matrix that separates these cells, and this is so that they can bear weight, withstand physical force, and also endure abuse. Again, their job description is to what? Protect and bind the body, and they do it very well as a result of their extracellular matrix secretions. Okay, if we look at the structural elements of connective tissue, and here we're thinking mostly of connective tissue proper, this would be ground substance as well as fibers embedded within the substance and the cells that make the extracellular matrix. The composition and arrangement is going to vary in different connective tissues, and we'll look at a few of them in more detail. You'll see, for instance, in dense regular connective tissue that the fibers have a very regular parallel arrangement, as do the other components of the extracellular matrix, whereas in irregular connective tissue, the fibers are a little bit less organized, and as a result, are a little bit less able to withstand shear stress and tearing stress. Um, the cells, of course, produce the extracellular matrix, and the ground substance serves as essentially a medium in which these fibers can effectively do their job, in addition to providing now a barrier to penetration. Ground substance is essentially an unstructured material that fills the space between cells, and it's also a medium through which we can have diffusion take place between the capillaries and the cells. And this is how a lot of the connective tissue cells manage to survive, to get energy in order to continue to live, is by diffusion of oxygen and nutrients through this extracellular matrix in order to supply now the needs of cells like fibroblasts and chondroblasts, and to a lesser extent the other cells that you find in connective tissue, such as lymphatic cells and nerve cells. The components are interstitial fluid and cell adhesion proteins, which essentially seal the cell to the surrounding matrix. Proteoglycans make up the bulk of the molecules we find in this matrix. It's a very unusual looking conglomeration of proteins and sugars that are very good at trapping water and are they have a profound negative charge, which means that they can resist compression because they very easily spring back to their original shape. Chondroitin sulfate and hyaluronic acid are two of the sugars that we find associated with the protein core, and in those sugars we find again a lot of negative charges associated with the monosaccharide residues. It traps a lot of water primarily due to its charge, and this affects the viscosity of the ground substance. And for those of you who don't know what viscosity is referring to, this is basically the resistance to flow. This is essentially the way of saying that the extracellular matrix is a very thick substance, and as a result tends to hold its shape. There's three types of fibers that provide support in connective tissue. Collagen is the strongest and most abundant type. It is tough and it provides tensile strength, so it resists tearing. Elastic fibers, of course, have the ability to stretch and recoil, and reticular fibers are short, highly branched collagen fibers that have a slightly different set of residues in them than collagen proper. They branch and form networks that have a little bit more ability to stretch and recoil than just straight up collagen. Okay, so we tend to find reticular fibers a lot more in loose connective tissue than we find in dense, regular, or irregular connective tissue. The cells that are tasked with producing and maintaining the matrix are scattered throughout. And if you look at connective tissue under a microscope, one of the things that you find is that there are very few cells and a lot of space between them. Blasts are cells that are immature initially and mitotically active, meaning they divide quite a bit. And they are what secrete the ground substance and the fibers through active transport. The fibroblasts are found in connective tissue proper, while chondroblasts are located in cartilage, osteoblasts in bone, hematopoietic stem cells in red bone marrow, and cytes are the basically grown-up derivatives of these blasts. Once they've deposited the matrix and get trapped inside it, chondroblasts, for instance, will become chondrocytes, and osteoblasts will become osteocytes. The fibroblasts will continue to secrete the extracellular matrix, and so they don't become, there's no such thing as a fibrocyte per se. At least I haven't seen anything like that in the literature. Okay, other cell types that we find in connective tissue are adipose cells or fat cells that store a lot of triglycerides, so that's a nutrient store for the body, as well as, of course, providing padding and protection. Also, white blood cells, which we know better as leukocytes, which are our body's defense against disease. White blood cells can be broken down into two broad groups: the granulocytes, which are the eosinophils, the basophils, and the neutrophils, and the agranulocytes, which are the monocytes, which eventually become macrophages, and the lymphocytes. And this distinction, granulocyte versus agranulocyte, refers simply to the presence of staining granules in the cytoplasm that are filled with proteins that are chemical defenses against the pathogen. In the granulocytes, we can very easily see these granules. They have very distinctive colors. And in agranulocytes, the granules are there, but they're more difficult to see. In fact, they're almost invisible. And again, these cells are designed to protect us against disease, so they rush to the site of injury and attempt to kill any pathogen that tries to penetrate. Mast cells initiate inflammation through the production of histamine, which they generate in their cytoplasm and then excrete. The histamine's job is to promote inflammation. You probably are familiar with inflammation. This is what happens when an injury swells up, turns red, gets warm, and hurts. And this is all designed to bring blood to the area. The blood contains, of course, the cells that are going to fight the pathogen and also the cells that are going to rebuild the tissue. Macrophages are basically eaters. They run around inside living tissue, and they eat dead cells, and they also eat pathogens and infected cells. And when they do that, they take the little bits of the pathogens or the infected cells and they display them to the immune system as a warning that there's a pathogen inside the body that's attempting to cause disease. And this results in the selection from the lymphocyte population of B and T cells that are specifically designed to fight that particular pathogen chemically and physically. And here's just a look at what some of these different cells look like. This is loose connective tissue, areolar tissue, and you can see a lot of these different cell types as well as the extracellular matrix components. The macrophages, which are the eaters, the fibroblasts, which make the extracellular matrix, the lymphocytes, which along with the macrophages coordinate the defense against the disease, fat cells, which store nutrients and provide padding and protection, mast cells, which secrete histamine and promote inflammation. Neutrophils are granulocytes, so they are a type of leukocyte, and they initiate chemical attacks against pathogens without necessarily having a tailored attack to that particular virus or bacteria or fungus. So they're sort of like our we would call them our innate immune system. The neutrophils, the eosinophils, and the basophils use the same strategy regardless of the identity of the pathogen, so that's why we call them our non-specific defenses. And of course, you're going to have a blood and nerve supply running throughout this tissue as well. Okay, now as we break down the different types of connective tissue, we're going to start with a type that's known as connective tissue proper. All connective tissues except for bone, cartilage, and blood are considered to be in this group. And there's two subclasses: loose connective tissue, which is either areolar, adipose tissue, or reticular tissue, or dense connective tissue, which is also known as fibrous connective tissue, which can either be dense regular, dense irregular, or elastic. Basically, these phrases refer to the organization of the extracellular matrix when we're looking at loose connective tissue. Areolar is going to have a more disorganized extracellular matrix with a lot of collagen fibers, a lot of reticular fibers, and elastic fibers running in all different directions. Adipose tissue is characterized again by the presence of large amounts of triglycerides, which are present inside the adipose cells, known as adipocytes, as a giant fat droplet. And then reticular connective tissue is going to have a lot of web-like fibers running in various directions. Dense connective tissue looks histologically quite different. It has usually a rather regular array of extracellular matrix proteins. We find a lot of these in tendons and ligaments. Dense irregular connective tissue is a little bit more rare to find in the body. Elastic connective tissue is found in areas that are subjected to distension and recoil. So let's start with areolar connective tissue to begin. This supports and binds other tissues. It is thought to be a universal packing material between tissue, and it's the most widely distributed. It provides a reservoir of water and salts. It defends against infection. It stores nutrients in the form of fat. It contains fibroblasts, which secrete and maintain the matrix, and a loose arrangement of fibers embedded in what we know as ground substance. When inflamed, it soaks up fluid, and this is a phenomenon known as edema, which is a hallmark of inflammation. It is one of the ways that we know that there's fluid coming to the area of the injury, and this also produces pain, which causes us not to use the affected area as much and give it a chance to heal. Adipose connective tissue we know as white fat. It's similar to areolar tissue, but it has a lot of nutrient storage. The main cell type is the adipocyte. That literally means fat cell. It stores nutrients. It doesn't have much extracellular matrix, so in that sense, it's a little bit different from most other connective tissues. It has a lot of blood vessels, so it's richly vascularized, and the reason for that is that the fat that's stored in these cells is used as nutrients for other tissues, and so we need to have proximity between the adipocytes and the circulatory system so that we can occasionally burn that fat in order to generate energy between meals. It's important in shock absorption and insulation as an energy store. Brown fat is an unusual type of adipose tissue which essentially contains triglycerides, but it also has mitochondria, which are the energy-producing organelles of cells that have a metabolic pathway called the electron transport chain, designed primarily to produce heat instead of to generate usable energy for the body. So this is one of the ways that the body maintains its temperature. It's a thermogenic tissue. It uses these lipid fuels to heat the bloodstream. An example of where you'd find brown fat are between the shoulder blades of infants, and we also find it in animals that do hibernating, such as in bears. This helps to maintain their body temperature when they hibernate. Reticular connective tissue resembles areolar tissue, but the fibers are what we call reticular fibers. The fibroblasts are termed reticular cells. Examples of reticular connective tissue are found in places such as the lymph nodes and the spleen, where they support free blood cells. We also find this type of tissue in the bone marrow. Okay, so areas where we find formed elements of blood, and also areas of hematopoiesis are populated by this reticular connective tissue. Next connective tissue we want to talk about is dense regular connective tissue, and this is tough stuff. It's made up of closely packed bundles of collagen fibers that run parallel to each other to the direction of which force is produced. They're white structures that have great resistance to pulling and shear stress. The fibers are a little bit wavy, so they do have some stretch, some elasticity in them. The fibroblasts manufacture the fibers in the ground substance. There tend to be very few cells in these tissues, and it is usually poorly vascularized. And this is one of the reasons why tendons and ligaments take a long time to heal, because of the poor blood supply. In fact, when you injure a tendon, it usually takes several weeks for it to knit, whereas if you rupture a ligament, it usually has to be surgically repaired in order for it to restore function. Dense irregular connective tissue is made of the same elements as dense regular connective tissue, but the collagen bundles are thicker and more irregular, as you can see in the micrograph here. It resists tension in many directions. We find this type of tissue in the dermis and in fibrous joint capsules, which is the outer covering of the structure that encloses most synovial joints. It's superficial to the serous membrane, or the synovial membrane rather, that produces the synovial fluid that lubricates the joint. We also find it in the fibrous capsule of certain organs. But you can see here how this tissue looks a little bit different than dense regular. Elastic connective tissue has the ability to stretch. These connective tissue are found in vertebrae and in the larger arteries in the walls. So you can see here a cross section of aorta showing you elastic connective tissue. In the great vessels, this allows rebound, and so this sort of dampens out pressure changes which otherwise would damage the vessel. So it's incumbent on these structures to be able to expand and contract in order to ease some of the physical stress and strain that they go through. The fibers tend to have a somewhat regular arrangement, as you can see. Cartilage is a fundamentally different type of tissue. The chondroblasts are the cells that secrete the extracellular matrix of cartilage, and once they become trapped inside this matrix, they're termed chondrocytes. This is a tough but flexible tissue that lacks innervation. It's up to 80% water but can rebound after compression, primarily due, number one, to its high water content, but number two, to the fact that there's profound negative charge in the molecules that make up the extracellular matrix of cartilage. It's avascular, so it gets its nutrients from the membranes that surround it, and also from the fluid that the cartilage is bathed in. For instance, synovial fluid contains nutrients that keep the chondrocytes happy and healthy, as well as oxygen. The perichondrium is the living membrane that surrounds the cartilage and is the source of the chondroblasts, which also eventually become the chondrocytes. There's three types of cartilage: hyaline, elastic, and fibro, and we'll talk about each one of them here. This is an example of hyaline cartilage. We find this at the ends of bones and in the epiphyseal plates. It's amorphous. It contains lacunae with chondrocytes embedded in them, but they're nourished primarily by the fluid and the membranes that surround them. The chondroblasts produce the matrix, and when they mature, they get essentially restricted to this very small physical space. And this is the material actually that the skeletal system is made out of before it mineralizes, and by the time the newborn emerges from the birth canal, all the hyaline cartilage has been replaced by mineralized bone. It's important in reinforcing and supporting bodily structures. It's a cushioner, and it resists compression stress. We also find this type of cartilage in the costal cartilages, which you can see here connect the ribs to the sternum. Okay, we find it also in the trachea and the larynx. Elastic cartilage has the ability to stretch. It's similar to hyaline cartilage, but it has more elastin in the matrix. helps to maintain the shape of a structure while allowing it some flexibility. We find this in the epiglottis and in the external ear. And if you look at the micrograph here, you can see the elastic fibers running through the extracellular matrix. Fibrocartilage is found in places such as the outer covering of the intervertebral discs, where it forms a ring-like structure called the annulus around a liquid center. It has a matrix that's similar to hyaline cartilage, but it's less firm. There's a lot of collagen in the extracellular matrix. It absorbs compression and it resists shock. We also find this type of tissue in the pubic symphysis, which is the joint between the anterior portion of the coxal bones, and we also find it in the menisci that are in the knee joint. Meniscus is basically a cartilaginous structure that increases the goodness of fit between the distal head of the femur and the proximal head of the tibia. Okay, bone is a very important connective tissue. It's also known as osseous tissue. It supports and protects body structures and stores fat, especially in the long bones in adults, and synthesizes blood in the red marrow in the flat bones in the adult skeleton. It has more collagen than cartilage does. It has a lot of inorganic calcium salts that resist compression force. The osteoblasts are the cells that produce the matrix. The osteocytes get trapped inside that matrix. They were once osteoblasts and they help maintain it. And the osteons are the structural units of compact bone. And then there's another cell type that we should be aware of, which is the osteoclast. The osteoclast. Let's write that down here in blue. Okay. And the osteoclasts come from a totally different lineage. Osteoclasts, that should be a C. They're multinucleate cells, and these are bone dissolvers. So they work in conjunction with the osteoblasts to remodel bone. An osteon, you can see here, I'll draw a circle around an osteon here in blue. This is an osteon here. Okay, and there is a central canal through which an artery, a nerve, a lymphatic, and a blood vessel run. And then these little things here that I'm drawing circles around, these are the osteocytes, and they're trapped inside lacunae just as chondrocytes get trapped inside lacunae. And you can see these tiny extensions that radiate out from the osteocytes. These are cytoplasmic extensions that protrude through canaliculi, these tiny hollow spaces between the osteocytes, and this is what allows the osteocytes to exchange waste products and nutrients until they are transported to a cell that lies near a blood vessel, at which point we can either drop off waste or pick up nutrients as the need arises. So I'm just circling here the osteocytes. Okay, you can see them here. And if you look also very carefully, you can see what we call concentric lamellae, and let's outline those in red. They're a little tough to see in this picture, but if you can imagine that this mineralized matrix gets deposited in layers just like the rings on a tree, except in bone the youngest rings are on the outside and the oldest rings are on the inside. But you can see here these concentric lamellae. Concentric just means shares the same center. Okay. This is richly vascularized tissue. It needs to be because bone as living tissue is constantly responding to physical stress, changes in weight and force, and as those changes occur, the bone has to increase its density along the lines of applied force while reducing density where the force is less frequently applied. And so the osteoclasts and osteoblasts together help get that done. Blood is one of our two liquid connective tissue, the other being lymph. It's an atypical connective tissue because it's a fluid. Red blood cells are the most frequent blood cell type, and they carry oxygen and carbon dioxide to a lesser extent. They also contain leukocytes, which we call white blood cells, and platelets, which we call thrombocytes. The fibers are soluble proteins that precipitate during blood clotting. You normally can't see them unless the blood vessel has been injured, at which point collagen fibers trigger a series of chemical reactions in the plasma that ultimately result in the formation of a fibrin clot. And it's important in transport. Transport of what? Oxygen, nutrients, waste products, hormones, fluids, and electrolytes. All of these are carried by the blood. Okay, muscle tissue is special because it's highly vascularized and it's responsible for movement. It's able to take chemical potential energy and turn that into kinetic energy with the loss of heat as a byproduct. And there's three types of muscle tissue we need to become familiar with. Skeletal muscle tissue, which is found obviously in skeletal muscle. It is voluntary and also has a feature known as striations, which you can see when you put skeletal muscle tissue under a light microscope. You see light and dark vertical bands running perpendicular to the long axis of the muscle cell. Cardiac muscle tissue is found exclusively in the heart, and it is under involuntary control. There are cells embedded in the heart that are called pacemaker cells that tell the heart when to beat, and those pacemaker cells interact with the autonomic nervous system, which can speed up the heart when activity levels pick up and slow the heart back down when we're at rest. It is also striated. Smooth muscle tissue is non-striated and involuntary. We find it in the walls of hollow organs other than the heart, and it still is capable of contraction, but it is the slowest of the three muscle types in terms of producing movement. And so here you can see what skeletal muscle looks like. This is a micrograph of skeletal muscle. It's found attached to bone via tendons, and these light and dark bands that you can see, those are the striations, and those indicate the areas of overlap between a muscle protein called myosin and another one called actin. You can also see that there are a lot of nuclei per single cell. Muscle cells are unique in that the way a muscle cell forms when it's derived from mesoderm is that the cells that are going to be muscle cells gather together in a group, and then they lose their intervening cell membrane and become this long cylindrical cell with many nuclei whose job primarily is to make muscle protein so that it can produce movement. There's also quite a few mitochondria in muscle cells because they need to use a lot of energy in order to generate movement, and an extensive endoplasmic reticulum which is filled with calcium, which is what triggers muscle contraction inside the muscle cell when it experiences an electrical signal that is transmitted to it from the nervous system. And so there you can see the photomicrograph of skeletal muscle. Now, cardiac muscle is very similar. It's found only in the heart. It is striated and involuntary, one nucleus per cell, and it has a lot of branching in it. And it features these unusual structures called intercalated discs. You can see them here, 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 and so on. And these intercalated discs represent cell-cell junctions, which are populated both by gap junctions, which are a type of communication junction, and desmosomes, which are a type of tight junction. So the desmosomes hold the heart together as it beats so that it doesn't come apart every time it pumps, and the gap junctions allow charged particles to move very rapidly between muscle cells, and this is what helps the electrical signal move across the walls of the heart so that it can beat in a coordinated fashion, where the atria squeeze first after they receive blood from their respective organs, the right atria receiving blood from the body and the left atria receiving blood from the lungs, and then that's followed by the ventricles squeezing. The ventricles are termed the discharging chambers. They're the lower chambers of the heart shown here. The right ventricle sends blood to the lungs, and the left ventricle sends blood to the aorta, which leads to the rest of the body. And if we didn't coordinate this pumping action, all the heart would do is shimmy, and that wouldn't be an effective way to move blood around the body. So it's these gap junctions in combination with this electrical tissue that's inside the heart that coordinates the pumping action of the heart. Now, smooth muscle looks very different. Smooth muscle has one nucleus per cell, and the cells are spindle-shaped, and they're found primarily in the walls of hollow organs, in blood vessels, and in lymphatic vessels. And they produce internal movement. They move substances, for instance, through an organ, or help to mix substances in an organ, and they help in functions such as moving the blood and lymph around the body and moving food through the digestive tract. Um, these cells are the slowest of the three muscle cell types in terms of contraction. They're under autonomic nervous system control, but they're still very effective at what they do, which is primarily generating internal movement in the body. Now, nervous tissue is very, very different. Nervous tissue looks like no other tissue in the body. It's the component of the nervous system, and that is made up of the brain, the spinal cord, and the nerves. It regulates and controls body function, so it helps to maintain homeostasis. It's one of the body's two major communication networks, the other one being the endocrine system. Neurons are the cells that actually receive and generate electrical signals, and they are assisted in this task by cells that are called neuroglia, which you could think of as the nerve cell's personal assistants. They support the cells that send and receive the electrical signals. They also insulate and protect them. Now, the other aspect of tissues that we need to address before we end this chapter is this idea of membranes that line the body. There's at least two primary tissue types that make up what we think of as a tissue membrane. So don't confuse this with a cell membrane. An epithelial layer that's bound to an underlying layer of proper connective tissue. These are termed simple organs because, again, an organ is defined as a combination of more than one tissue performing a task. And there's three types of membranes: cutaneous, mucous, and serous membranes. Cutaneous membranes have a keratinized cell type that covers the outer surface, made up primarily of stratified squamous epithelia attached to an underlying layer of thick connective tissue, which we term the dermis. It is a dry membrane, and it forms a barrier between the inside and outside of the body. It's a barrier to water penetration, it's a barrier to infection, and it also serves as a sense organ, a place where vitamin D is synthesized, and it's also important in regulating our body temperature, just to name a few of its functions. Mucous membranes line the surfaces of body cavities that have openings to the outside world, such as the digestive, the respiratory, and the urinary system. The mucosa indicates the location, but not really the cell composition. They're characterized as moist membranes that are bathed by secretions, or in the case of the urinary system, bathed by the urine. The epithelial sheet lies over the connective tissue layer, which is called the lamina propria, and in many cases it secretes mucus, such as in the case of the digestive and the respiratory system. The mucus, remember, has a different role in each of these systems. The mucus in the respiratory system is there to trap particulates so that the cilia that line or that are on the apical sides of the epithelial tissue in the upper respiratory can move that particulate-laden mucus up towards the oral cavity, or we can either spit it out or swallow it. And the mucus in the digestive system is there to protect the lining of the digestive system and to lubricate the food. Now, serous membranes line internal body cavities that are not open to the outside world, and they produce a fluid called serous fluid, which is a lubricant. We find a lot of these membranes in the ventral body cavity. An example would be the pleural and pericardial membranes and the peritoneal membrane. It's made up of a simple squamous epithelial layer which rests on areolar connective tissue, which is relatively thin. The parietal serosa are going to line the internal body cavity wall, while the visceral serosa covers the internal organs, and there's serous fluid between the layers, and that's what provides a lot of the lubrication. These membranes are moist, and again, they're pictured in the diagram. You can see the pleural, the pericardial, and the peritoneal membranes all pictured in the diagram you're looking at. Now, occasionally what will happen is the tissue will become damaged and it'll need to be repaired. This is necessary when barriers are penetrated because this represents a port of entry for pathogens. So cells will divide and migrate to the area. This phenomenon occurs in two major ways: through regeneration, where you generate the same kind of tissue to replace the old tissue, or in fibrosis, what we do is we produce scar tissue to perform make us basically a temporary seal where the wound was, and we lose the original function. So in scarring, you don't get back tissue that looks and acts the same as the original, whereas in regeneration you do. And so different tissues are capable of different types of tissue repair. If we look at the skin, what we're looking at here is an example of a laceration. You can see here the cut. The inflammation is going to set the stage for this process. It causes the release of inflammatory chemicals, followed by the dilation of blood vessels, which increases the vessel permeability and will eventually lead to the formation of a blood clot. Remember that blood, when the flow slows down, will tend to form a clot. And that clot is a temporary seal so that we can hold on to our blood, which we need in order to live. In addition to that, we also are going to attract to the area of the wound white blood cells, lymphocytes and macrophages, which are going to destroy damaged tissue and also battle any pathogens that are trying to get into the body through the wound. As we move on through the repair process, organization eventually restores the blood supply. The clot is then replaced with what we call granulation tissue, and the epithelium begins to regenerate underneath where the wound is, while the fibroblasts make collagen fibers that serve as a framework to bridge the gap, and debris is broken down by the phagocytes that are attracted to the area, due again to the secretions of the damaged tissue and also to the actions of histamine. And then finally, in the last stage of tissue repair, we see regeneration and fibrosis. The scab will detach, the fibrous tissue will mature, the epithelium will thicken and begin to resemble the adjacent tissue, and this results in a fully regenerated epithelium with some underlying scar tissue. Different tissues have different capacities for regeneration. Some regenerate extremely well. These include epithelial tissue, bone and areolar connective tissue, as well as dense regular connective tissue and hematopoietic tissue. Others have moderate regenerating capacity. This includes smooth muscle and dense regular connective tissue. And then those with virtually no regenerative capacity include cardiac muscle and nervous tissue in the brain and spinal cord. Although new research shows that under certain circumstances cell division can occur, but this is relatively rare. There are efforts underway to try and trick these tissues into regenerating so that we can more effectively repair damage to these tissues when they occur in human beings. Now, the last couple of slides talk a little bit about really what we started the lecture off with, which is the development of the three primary germ layers. And what you're looking at here is just a better drawing of what I was trying to show you earlier. You can see here the embryonic disc, which forms from the inner cell mass of the blastocyst at about 16 days. And what you're looking at are the three germ layers. You can see here the ectoderm, the mesoderm, which comes from cells that migrate through the primitive streak and populate this area between the ectoderm and this lower layer, which is called the endoderm. And you can also see the types of adult body structures that each of these tissues forms. You can see that the ectoderm forms the epithelia and the nervous system, as well as some connective tissue. The mesoderm forms the cardiopulmonary and musculoskeletal system, and also some connective tissue. And then finally, the endoderm forms the inner lining of the digestive system. And the reason that these cells are able to perform these particular tasks is that the information in the nucleus, as these cells develop through time, becomes more and more restricted to the point where by the time a cell is an ectodermal cell, it can only read from the ectodermal handbook in the DNA. The same goes for the mesodermal and the endodermal cells. Even though the information is still in the nucleus, it's basically sealed off. And then as these cells continue to generate these other adult tissue types, we even more radically restrict the areas of DNA they're allowed to access to control their behavior. So for instance, by the time a muscle cell is a muscle cell, it's only reading from the muscle cell handbook. The skin cells are only reading from the skin cell handbook, and so on. And this is going to have profound effects on the cell shape, the products that the cell makes, and what the cell does in the living system. As we age, we lose a lot of this function. Normal function usually works quite well through youth and middle age if we have adequate diet, adequate circulation, and infrequent wounds and infection. The epithelia, though, begin to thin as we get older, and so they're more easily damaged, and tissue repair is less efficient because there are fewer cells around to affect tissue repair, and this is due simply to cell death. Bone, muscle, and nervous tissue begin to atrophy, and that's simply death due to disuse, or more probably accurate, death due to loss of information that's vital to keeping the cells alive. We talked in class about the fact that one of the most avidly championed mechanisms of cell death is the result of the loss of DNA information through successive cell divisions as these cells go through many rounds of mitosis, and eventually the feeling is that we'll eventually get to ruining vital information that the cell needs in order to live, and when that happens, the cell basically checks out, undergoes apoptosis, and then is no longer part of that tissue population. Also, as a result of increased lifespan, there's more likely to be cancer events that can establish themselves and escape detection by the immune system and potentially generate the disease. And so this is simply a matter of, if you live long enough, eventually those mutations that take place in DNA every day, a few of them may not get caught, and then the likelihood that they'll begin to travel around the body in the process of metastasis becomes more and more likely. Okay, make sure and review this material in preparation for the quizzes and the exams, and I will see everybody in class. Have a good day.